what? One chromosome. So this is what happens in the first stage in both meiosis and mitosis. We got 46 chromosomes, right? In interphase, what happens to those chromosomes? They go through DNA replication. So now those 46 chromosomes will be doubled, right? So imagine now 46 of these little X's, right? That's what we get in both interphase of mitosis 1, or mitosis rather, and then meiosis 1. We always double at the first stage. Always, 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 always. The difference is, in mitosis, we took each of these 46 chromosomes, so remember, 46 X's, right? And we lined them up, didn't we? In metaphase. For mitosis is what I'm talking about. And then what did we do? We separated those 46 X's in anaphase, right? So if we separate, we got 46 of these lined up, just like this. Right now you see two X's, but there should be 46 of them lined up in metaphase. If we separate them all, that means that 46 chromatids went over here, and 46 chromatids went over here, right? That stayed where that cell was 46, or excuse me, 23. Sorry about that. I didn't mean to say 46, it was 23. 23 go one way, 23 go the other way. 23 stay over here making one cell, 23 stay over here making another cell. Right? Well, no, let me go back. I did screw it up. Let's erase that. That was meiosis. Let me start over, I'm so sorry. The difference was we had 46 lined up. Let's scratch what I just said. 46 lined up, so now 46 chromatids go this way. Y'all see that? Are we all good? And 46 go that way. That means that now we have, what, 2N or N over here? 2. We have 2N, making a full diploid cell. That's mitosis, right? The difference is that in meiosis, here's where we want to start getting it. In meiosis, we take these replicated chromosomes, instead of lining them up like this, what do? We line them up like this. So now what's this known as? A tetrid. Right, so with tetrids occurring in prophase now, right, because where did the replication take place? Interphase. In interphase. The replication meant now we have a sister chromatid attached, right? Now in prophase 1 of meiosis 1, we get tetrid formation, right? They stick together, and what do they start doing? They start crossing over. So the little tips where you see them touching start exchanging genetic information. So what this tetrid is made of is what? Homologous chromosomes that share the same trait. So now we're looking at 23 chromosomes from mom, right? 23 chromosomes from dad, mixing together, finding similar traits, that's what a homolog is, coming together in prophase and crossing over. So this is what metaphase looks like in meiosis 1 compared to mitosis. See the difference? We get tetrads lining up. So how many tetrads are lining up?
2.
Well, you see 2, but how many should it be? 23 tetrads lining up, right? Okay, so now the purpose of Anaphase 1 is what? To separate the tetrad, right? So remember, let me go back. Let's clear everything up. I'm so sorry I screwed it up. This red represents 23 X's from mom. This blue represents 23 X's from dad. Okay? Right now, all of our cells have 23 from mom, 23 from dad, right? All we're doing now is sharing similar, we're finding similar chromosomes, because we all have a duplicate copy in our cells, right? We're lining them up. Now in Anaphase 1, we separate the tetrads. So if this represents 23 from mom and 23 from dad over here, when we separate, what will this cell become?
Haploid.
Right, that's where we get the haploid. So the purpose of meiosis 1 is to get down to the haploid number. Even though it looks like an X, it's still only 23 of them. So the products of meiosis 1 is haploid, right? Okay. 2 haploid cells in meiosis 1, and for meiosis 2 it changes to 4 haploid cells. That's the takeaway. That's what you really, really, really need to know. Okay. I'm not as proficient with the pipe cleaners as I used to be. Okay, so let's look at the difference between meiosis 1 and 2. Meiosis 1 told us that we had tetrads lining up in metaphase, so that when we split them in anaphase, the 23 from dad and the 23 from mom will separate, right? Having already been crossed over. So now we have genetic variability on this chromosome, don't we? Imagine now it's red with little blue tips, randomly. And this one would be blue with what? Red tips, just randomly, right? So the 23 from dad, now with a little bit of information from mom, right? Divide. So now we have a haploid cell over here and a haploid cell over here. These 2 cells are going to be what we start meiosis 2 with. Right? So now, this one on its own, right, will go through its own meiosis 2 cycle. Right? So how many of these will be? It's 23. So now we got 23 chromosomes lining up, right, in meiosis 2, in metaphase, right? There is no second interphase, by the way. We don't double the chromosomes again. There really is no interphase in meiosis 2. Okay, they call it interkinesis, if anything. But we basically skip over interphase, in a sense, in meiosis 2. There's no doubling. There's still a stage, but there's no doubling of genetic information. So now taking this one haploid cell, right, from meiosis 1, we start meiosis 2. Now how many we start with? 23, these little X's. Now what do we do in metaphase? Line them all up. What do we do in anaphase? Split them, right? So this one 23 chromosome cell will now split into 2. Chromatid here, chromatid there, right? And now these will split, right? Giving you 4 unique. So that's how we get the 2 haploids in meiosis 1 and the 4 haploids in meiosis 2. All right. So I don't know if that helped at all. Sometimes, honestly, I know how this goes. Looking at meiosis, the more you look at it, the more confusing it gets, truthfully. But by the time we finish it, you'll see what's what. Now, where is our meiotic cycle here? Is it coming up in the PowerPoint? It must be. Okay, well, let's get through these few slides and we'll get to the pictures. Okay. So the site of fertilization is where? In the fallopian tube. If we get this egg, of course, coming out, a mature follicle releasing the egg. If a sperm is in the neighborhood, we get fertilization in the uterine tube, hopefully. Although there are occasions where the egg can be coming outside the uterus, outside the fallopian tube, and somehow crazily being fertilized outside the uterus or outside the reproductive tract. That would be kind of crazy, wouldn't it? But it has happened. That would be an ectopic pregnancy when it's fertilized outside and somewhat carried to term. I don't know how that happens, but it has happened. Yeah, and sometimes just into the peritoneum. But I don't know how in the world it finds nourishment. I've never researched it enough to find out. Okay, the 3 layers of the uterine wall, myometrium would be what? Muscle, endo, is basically that mucosal lining, right? The simple columnar. If development of endometrium starts occurring outside, the uterus, the endometrial cells can go elsewhere. That would be considered endometriosis. Perimetrium on the outer side. The 2 layers that we're going to concern ourselves with as the menstrual cycle comes around is the functionalis and the basalis. This one is the one that is shed, the functionalis, that is shed during those first, major, days 1 through 5, let's say, of the menstrual cycle. Which always leaves us with the basalis layer that would form the new functionalis layer. Okay, so there's the basalis, where the deeper arteries are. This is all what basically breaks down and sheds off during the days 1 to 5 or 1 to, sometimes even 1 to 8 of the menstrual cycle. There's our two anatomical terms for the cervix. Stratified squamous throughout the vagina with some small amounts of rugae. It's basically called the birth canal. And then the outer anatomy of the female here, we mentioned the major and the minor. Okay. Just as in the male, erection of the female tissue by parasympathetic flow and orgasm by sympathetic flow. I'm not sure why I wrote that. I guess it was in the book somewhere. That's not going to be on the test, of course. What two hormones did control milk letdown? Oxytocin and prolactin. Great. Okay, that's about all we would need to know for that. And the milk passing through these lactiferous ducts, which open to the outside. So that being the one major part, that and the areola around the nipple, being the two major parts that you would have to know for the breast tissue. The lactiferous ducts. Lactiferous sinus is just a collecting area of the milk before it passes through the nipple. And just general anatomy, if you've not known this, of course, if you don't have breasts, you probably would not know this, but the muscle tissue, the pec major muscle, is always behind the breasts. And it basically is just fat tissue and glands making up the breast tissue. And what are these here?
Ribs.
Just the ribs that are cut. Very good. Okay, so for meiosis again, maybe with these pictures it'll help. If you want to talk with me after class for a little bit and go through the pipe cleaners again, it helps some people, it doesn't help some people. So if you want me to run through this with you, I can. I think the biggest difference I want you guys to realize is the difference between meiosis 1 and 2. Right? We have crossover and tetrad formation in meiosis 1. So meiosis 1 is by far more interesting. That's where all the special events are in meiosis 1. Meiosis 2 is really just kind of like mitosis in a way. It looks like mitosis, really. The different products, of course, but it looks the same as what you would kind of get from mitosis. Okay, so we always start, right, with diploid. The gametes that we produce are always going to be haploid. So when mom and dad, right, get together on a Saturday night at 6:30, right, after dinner and a movie. We want to romanticize this, right? Imagine this. My mom and dad, way back in the 50s, right? They could literally go out, go to the movie, get a hot dog or a hamburger or whatever they ate, I don't know. But they've told me this. They could do all that, a movie and a hamburger and french fry, for like $1 or $2. Right? Isn't that crazy? Like 50 cents to get in the movie for the both of them. It's a pretty good date, isn't it? Or a cheap date, rather. Not a good date. Right, $10 a week, exactly, yeah. So anyway, we can romanticize it like that. But I like to think of my mom and dad. They're so cute. They really are. My dad has been the only man my mom has ever known. The only man she's ever dated since like 15. Okay? Yeah, that's kind of crazy. She got married at 17 too. So that's nuts. Anyway, okay, so we get gamete formation. In meiosis 1, we have it from 2N to N. Why do